Hello, everyone, and thank you for joining. It's inspiring to see this growing global community continue to come together. Families, clinicians, and researchers all connected by a shared purpose. Each month, we come together because knowledge matters. For families navigating neuroblastoma, understanding research shapes real decisions, real timelines, and real hope. Today, we're focusing on a topic that represents one of the most exciting directions in the cancer research, liquid biopsy. Liquid biopsy has the potential to change how we monitor disease, how we understand response to treatment, and how we detect relapse earlier and more accurately. It holds real promise for improving how we care for children with neuroblastoma. We are truly honored to welcome Dr. Gudrun Schleiermacher, a leading expert in neuroblastoma research and a pioneer in the field of liquid biopsy. Her work has been instrumental in advancing our understanding on how we can use blood-based biomarkers to better guide treatment and improve outcomes. Today, Dr. Schleiermacher will help us better understand what liquid biopsy is, how it is being used today, and what the future may hold. For families, this knowledge is empowering, helping to better understand the tools that may shape the next generation of care. Dr. Schleiermacher, thank you so much for being with us today. We are grateful for your time, for your leadership, and we look forward to learning from you.
Thank you very much. Thank you very much for this invitation. It's an honor, a pleasure to be here today and speak about liquid biopsies. Thank you very much for the very kind introduction. Just to be sure, is my screen being shared? Is it okay?
Yes, that's fine.
I can't see the Zoom anymore for whatever reason. Apologies. So just to give a very brief overview so you know who I am a pediatric oncologist. I work in Institut Curie in Paris in France, and I have two activities. I'm involved in patient care. I see patients with neuroblastoma and also other solid tumors, and I'm also a delegate director for translational research in our integrated pediatric oncology center, and I lead a team of research, and we focus on liquid biopsies and the study of clonal evolution, and I'll explain a bit more what that is and what we can do in this respect. So to give a bit more about my background at Institut Curie in France. So this is really a state-of-the-art hospital that goes back to 1920, founded by Marie Curie, who had discovered radioactivity. And in this hospital, we have an integrated pediatric oncology center. We see approximately 380 new patients with solid tumors a year. And we also are linked to our research unit, which is led by Olivier Hérault. And in Europe, SIOPEN is our association that brings together clinicians and healthcare professionals in the SIOPEN network, founded in 1998. It has been at the origin of numerous clinical trials for high-risk neuroblastoma and also low and intermediate risk neuroblastoma, as well as patients in relapse. And within this structure, I lead the translational research committee, and the aim of this is really to streamline, coordinate our translational and fundamental research efforts. So today I want to speak about liquid biopsies, the hope behind this novel technology, and you will see that maybe it's not as novel as this, but we need to think about how to use it in the context of today. And I want to think about how we can use this in our overall aim to identify the best treatment for each patient with neuroblastoma. And should relapse occur, also how to use this to identify the best treatment in case of relapse. And especially if there is no standard treatment, how can we identify patients with persistent disease or identify relapse? Can liquid biopsies really help us to face these challenges? In this talk, I want to address these questions in three different parts. First, I want to speak a bit more about what are liquid biopsies? How does this actually work? What can we see? What can't we see? And then I want to speak about how liquid biopsies are already in clinical use for patients with neuroblastoma today, and then think more about how in the future these technologies might contribute to our understanding of mechanisms of disease evolution and resistance. So what are liquid biopsies? For this, maybe it is useful to first think about what the word biopsy actually means. So this comes from the Greek word bios, life, and opsis, sight or to see. And of course, the idea to look at body fluids, the blood, the urine, saliva, cerebrospinal fluid is not new, because doctors have been looking at this ever since ancient times to figure out the disease a patient might be suffering from. More recently, this term has been coined really in the concept of using novel molecules that can be detected in body fluids that may not have been detected by the more standard biochemical analyses. And these molecules can be DNA, cell-free DNA, and its circulating tumor DNA component. I will explain that afterwards, as well as other molecules, proteins, enzymes, hormones, etc. So in the context of cancer, we often think about liquid biopsies as something that might detect tumor-specific biomarkers, molecules in body fluids. The real advantage of the approach of liquid biopsy, especially for solid tumors, is that a blood sample might be considered as minimally invasive. It might lead to sequential analyses that would not be possible if we're thinking about tumor biopsies of a solid tumor. And in these liquid biopsies, we can look at different analytes, different parts that could be analyzed, circulating DNA or cell-free DNA, also circulating tumor cells, proteins, cell-free RNA, circulating immune cells, extracellular vesicles. For a patient, a child with a tumor or a cancer, we want to use these analytes to look for tumor-specific elements that are distinguishable from those coming from a normal tissue. Now, when we talk about liquid biopsies, nowadays we often think about this as the study of cell-free DNA. And interestingly, a brief reminder, cell-free DNA was actually discovered in 1948 by a French team, a decade before the actual structure of DNA was known. And this French team had seen that DNA can be found in the blood of patients. In this very old article, they reported this in patients with different diseases, liver cirrhosis, and also during pregnancy. And now we know that cell-free DNA is of particular interest in patients with cancer. So cell-free DNA refers to these small fragments of DNA that circulate freely in the bloodstream outside of cells. The key characteristics are that these are small fragments, 150 or 200 base pairs long. They can be found in blood, but also in urine, saliva, cerebrospinal fluid, and the half-life is really short. Now, where do these fragments come from, and why are they so short? This is because actually in a human cell that is typically 10 or 100 micrometers, a huge lot of DNA has to be packed inside the cell. 10 to the power 9 base pairs in DNA has to be packaged. And in fact, that's 2 meters of DNA that has to be packaged into 22 chromosomes into 10 micrometers. And that is achieved because this huge long strand of DNA is wrapped first around what are called nucleosomes, histone proteins, and then these are tighter wrapped around into chromatin to form the chromosomes. And when DNA is released into the bloodstream, the DNA is cut by enzymes and in fact corresponding to the structure of the histones. So one of these little fragments is actually the length of the DNA around one histone. So that is why these are 150 to 200 base pairs. So where does the cell-free DNA come from? Why is it released into the bloodstream? Cells, any cells in the body, can undergo mechanisms such as apoptosis, necrosis, or secretion. And following these processes, such cell-free DNA fragments are released into the bloodstream. I'm more talking about blood now than the other fluids of the body. And in the blood, then these fragments are present in the plasma as opposed to the cell pellet. Now, if a number of cells in the body are not normal cells, but tumor cells, a certain fraction of the cell-free DNA will correspond to the circulating tumor DNA proportion of the overall cell-free DNA. So it's important to understand that when we talk about liquid biopsy and anything that is being analyzed in the plasma, in fact, we're always talking about a mixture of molecules that come from healthy cells and molecules that come from the tumor cells. And it is our task when we are looking for signatures, anything that might come from the tumor DNA, to see what proportion is actually specific to the tumor DNA versus the normal DNA background. So we know that in the context of a patient with cancer, the circulating tumor DNA fragments, a fraction of the cell-free DNA, can be used to identify tumor-specific genetic or epigenetic alterations. For any patient with cancer, this is of interest because we can look for copy number changes, we can look for structural alterations, we can look for mutational signatures, we can look for mutations, single base mutations, and its allelic fraction. And this, in the context of cell-free DNA and ctDNA, is simply determined by calculating the part of the molecules that can clearly be assigned to the tumor versus all the normal molecules. So cell-free DNA and its use for cancer patients is not new. In this diagram, you can see again that in fact its discovery goes back to 1948. So that's quite some years ago, but it's over the last 25 years that the huge leaps in the technological aspects of sequencing have enabled us to use this knowledge for patients with cancer. This is because first, nearly 25 years ago, the human genome was sequenced, and that enabled a huge leap forward with all our sequencing technologies. Before, when we were looking for mutations, we could only do this one gene at a time, one mutation at a time. And this led to the development of what we call next generation sequencing, or massively parallel sequencing, with all novel technologies that enable us to go so much faster. So when we're studying cell-free DNA, we want to look for circulating tumor DNA with its specificities with regards to a tumor. And in that context, to think about what we can see or what we cannot see, it is important first to realize what numbers we're thinking about. Maybe it's of interest to remind everybody that a human body is actually composed of 10 power 13 cells, so a huge lot of cells. And a tumor is probably just a small fraction. So if we see a tumor on imaging on an MRI or a CT scan of 1 millimeter, that's probably already several millions of cells. On the other hand, one cell contains DNA, as I explained before, and in one cell, you just have 1 picogram of DNA. So when we're working on plasma to isolate cell-free DNA, we typically extract plasma from 10 milliliters of blood, so that will be 5 milliliters of plasma, and we often get a yield of 10 to 100 nanograms of cell-free DNA. And so if we know that one cell contains 6 picograms of DNA, this amount that we are isolating actually corresponds to 1,000 or 10,000 cells. So that is important because if we are looking for one mutation, one molecule in what we have isolated in 10 to 100 nanograms of cell-free DNA, it means we cannot go beyond 1,000 or 10,000 normal cells. Because we can't see beyond one molecule in the 10,000 or molecules that we're looking at. It's simply technologically not possible to look for a molecule that's not there. And so that determines a bit the limit of detection of what we can pick up of 0.1 or 0.01% when we calculate it in variant allele fractions. And when we try to go beyond that to be even more sensitive, we have to come up with other approaches. I'll speak about that a bit in the end. But the limit that we can see something is really determined by the volume of the blood we're looking at and the amount of cell-free DNA in this. Some years ago, a researcher published the quantities of circulating tumor DNA, again as a fraction of cell-free DNA. And in the graph on the top, you can see all the quantities that were determined in patients with different tumor types. And you can see in this first publication for neuroblastoma in the blue bar. It was not very high, it was not very low either. And so we see that indeed in neuroblastoma, cell-free DNA can often be isolated. So how does our workflow nowadays look like? Frequently, we will work on tubes that have been collected. In the past, we could even do our analyses on leftovers from a routine full blood counts on EDTA tubes. Now, often commercially available tubes are used simply because these stabilize the cell-free DNA for a longer period of time, so we don't need to spin down the sample immediately and separate it from the cells. And then once the plasma separated from the cells in the blood, we can use really standard extraction kits and extract the DNA, the cell-free DNA. And when we look at this in electrophoresis, so to look at the size of what we're seeing, we want to see small fragments of around 160 base pairs. If we see bigger fragments, this means that we're probably not working with cell-free DNA, but the DNA that comes from the normal cells, from the white cells in the blood. And if we're looking for something that's tumor specific, this is not really going to be helpful. Then if we're studying cell-free DNA, typical steps concern what we call library construction. So this really means, in fact, that what we're doing once we've isolated the DNA fragments is we just add little bits and pieces to the DNA parts, and then these little bits and pieces enable us to put them in a machine that we can call DNA sequencer. There are a number of different technologies for this. And following sequencing, we want to find the molecule that has the alteration that might be specific of a tumor. In this case, I marked a red star to show that that might be a mutation. And then bioinformatics analysis is crucially important because we want to find this alteration and calculate its frequency with regards to all the other molecules that are not from the tumor cell. And so there are different sequencing techniques. We can use techniques that are called digital droplet PCR. They're really sensitive. They don't need a lot of input DNA, but they need quite some time setting up, and we need to know the sequence that we're looking for. On the other hand, targeted sequencing techniques such as next generation sequencing, they also need to know a bit what genes we are looking for. And then there are the wider sequencing techniques, low coverage whole genome, where we look at the whole DNA, everything in fact, or targeting the exomes, that means the genes that are expressed, or whole genome sequencing at really higher depth. So as we go from the digital droplet PCR to whole genome, we cover more of the genome, we get wider, but then we probably cannot go so much in depth because of cost issues and material issues. And when we use these cell-free DNA analyses in pediatric oncology, I like to think about it in different phases. First, for a child with a new diagnosis or suspicion of cancer, we can use these approaches in the context of diagnosis, molecular diagnosis, maybe also to contribute to the evaluation of disease burden. Then a child will go through treatment and will be in follow-up. And during the treatment and follow-up, these technologies might be used for the follow-up, maybe detecting relapse. If relapse does happen, these techniques might again be useful to look for molecular alterations that might guide treatment within the different precision medicine programs. This graph was drawn not for neuroblastoma specifically, but for adult cancer. And essentially, it means that we might want to think about our analyses and quantification of cell-free DNA in light of what might happen with regards to the overall tumor burden, leading to clinical diagnosis, treatment that will decrease the tumor burden, and should relapse occur, the new tumor burden, and maybe the cell-free DNA in some way might correlate with this, but we don't really know how this might correlate yet and what factors might influence the quantity of cell-free DNA. I think that it is probably the tumor burden, but also genetic features of neuroblastoma. MEK-amplified tumors probably have different cell-free DNA quantities than others. And so what we have looked at specifically in pediatric cancers is the overall cell-free DNA quantity in different tumor types. You can see neuroblastoma on the left, and neuroblastoma patients indeed often have higher cell-free DNA quantities than other pediatric cancer types, as you can see on the graph here. The red line indicates the cell-free DNA quantity that we often see in a healthy adult. And then we can look at this more specifically at the time of diagnosis. And in neuroblastoma, as you all know, we know that some biomarkers are of importance at the time of diagnosis to determine treatment. And these are typically in different collaborative groups, the MEK amplification status. Some collaborative groups want to look at the chromosomal copy number alterations and the mutational profile, in particular, the presence or absence of ALK mutations is also of importance. And so we have worked for quite some time together with other colleagues on whether we can determine these copy number alterations at diagnosis in cell-free DNA in neuroblastoma patients. And some of these publications go back to 10 years now. We were pioneer in showing that we can indeed look for copy number alterations, this example on the left, or mutations as indicated on the right in neuroblastoma patients at the time of diagnosis. However, things are not always so easy. In this example here, you can see the CT scan of a 4-year-old boy with a huge tumor next to the left kidney. This tumor actually does not show any capture of MIBG, and on the PET scan, you can see it lights up more in the periphery. The center is probably a bit more necrotic. So profiling of the tumor showed a number of alterations, for instance, the gain on 5p, which harbors the TERT gene, and other gains and losses. And upon sequencing, just a mutation in a gene that is not directly targetable. However, performing cell-free DNA analyses at the time of diagnosis, in addition, showed the presence of a mutation in the ALK gene at 1.9%, so lower than the actual mutation that was seen in the other gene that's present in the tumor. So this indicates that there's really some degree of heterogeneity with some mutation seen in the ALK, but not the other gene. And when we look at these sequencing results that we can obtain at the time of diagnosis using really real-world data, so this is not in any trial, we can see that cell-free DNA sequencing is feasible in probably around 80% of patients. And however, a number of patients will have non-contributive results. But over 60% will have contributive results at the time of diagnosis. So what about at the time of relapse? So at the time of relapse, as you know, in some instances, no standard treatment is available. And it is particular in these situations that it's really important to know the molecular profile so that we can propose the best treatment for a given patient. And it is thought that taking into account the molecular profile might be of interest. So a number of precision medicine programs have, during the recent years, published their results. And there's a general consensus that we can identify targetable or actionable alterations in over 2/3 of patients in relapse. And so what is particular here is that we know that at the time of relapse, new mutations might emerge that were not seen at the time of diagnosis. In particular, in neuroblastoma, we and others have shown that we see a high number of mutations, including ALK or other targetable genes that were not present in the tumor at the time of diagnosis. And so that highlights the importance of doing a new analysis at the time of relapse if we want to take into account sequencing results and molecular profiles for treatment decisions. And just to give an example, again, of a 10-year-old boy with a stage M metastatic neuroblastoma. You can see the widespread MIBG positive metastases in the spinal column and in some bones at the time of diagnosis. So this patient went through a number of treatment which did not work, which always led to stable disease. And this is some years back. And in fact, a bit by chance, we stumbled upon the cell-free DNA sample and did some sequencing on it, and then discovered this ALK mutation in the cell-free DNA. In fact, this was at a time when we did not know yet that this could be of relevance and interesting. But following this discovery, this patient was given a lorlatinib, an ALK inhibitor, and you can see that indeed this treatment worked because the bone metastases disappeared. The patient was in complete remission for over 3 years. Very unfortunately, then the disease came back and the patient passed away. But we do think that this was really the detection of the ALK mutation in the cell-free DNA that enabled this complete remission over several years. So what we have learned is that things are not so easy. And when we look at cell-free DNA at the time of relapse and compare it to the tumor, in some instances, we see a complete overlap of mutations. We call these SNVs, single nucleotide variants, if we don't really know if they have any biological function in the tumor or not. And in this instance, for one patient, 84 such events were seen, and they were completely identical between the tumor and the plasma. But then there are also instances where there is what we call heterogeneity, a number of mutations that are common to the tumor and the plasma, but also some that are specific either to the tumor or the plasma. And then this intriguing case where we saw many mutations in the plasma, but not the tumor, thinking that these probably came from a metastatic site somewhere. And so in our different precision medicine programs, what we have seen is that cell-free DNA sequencing at the time of relapse is successful in over 80% of cases and can lead to the identification of new alterations. And this is really important when we want to propose a targeted treatment. But what about the important question of disease monitoring? Can we use this not only to discover targetable alterations, but also to follow disease? And here first, if we just plot the cell-free DNA content over time, maybe a bit as expected, we can see that there does seem to be some kind of correlation between the cell-free DNA quantity, the ctDNA content at the time of diagnosis, if the treatment works, and then at the time of relapse. So a number of groups are now working on this and have firmly established the importance of using liquid biopsies and cell-free DNA analyses for tracking disease. Our colleagues in Germany have used these approaches, and in the example on the top, they have plotted copy number alterations and the ctDNA quantity. And we can see on the graph on the top how this correlates with the overall disease status. Our colleagues from SHOP in the USA have also used these approaches. And here again, as previously seen, the important correlation with the disease status, and also seen the emergence or enrichment of new SNVs, new mutations at the time of relapse, as indicated in these bars, with the yellow color indicating new mutations that appear at the time of relapse. And so if we look at this more in detail on the graph on the top left, the blue line indicates what we measure, so the variant allele fraction, VAF, for the ALK mutation in a given patient at different time points. And we can see that at 5 months of treatment, the VAF that had gone down to 0.2% went up to 4.5%. At that time, for the patient, a clinical stable disease was documented. But 4 weeks later, relapse was seen on MIBG. And so a number of colleagues have made similar reports now of cell-free DNA, evidence of disease progression or relapse before such relapse can actually be seen on an MIBG. Other things that we see when we look at cell-free DNA analysis more in detail is that, as I mentioned previously, we see new mutations at the time of relapse. And if we do whole exome sequencing, we can combine this knowledge together and then map out what we call clones that emerge at the time of disease progression or relapse. And we look at the pathways that these cellular clones target at a cellular level. And we think, as in the example here, when we see that regulation of cell differentiation or transmembrane transport might be impacted, that might be important also when we consider new treatments. Our colleagues, Dr. Bosse and his team, Dr. Burko and her team, have used these technologies to look more in detail for genetic alterations which emerge at relapse. And for instance, for patients receiving ALK-targeted treatment, they have seen ALK resistance mutations occurring in the ALK gene itself in 15% of patients, and also off-target resistance mutations in over a quarter of all these patients. And this is hugely important because what we want to know is what happens if a child who has been given an ALK inhibitor or other inhibitor treatment, if the disease does not respond anymore, if there's progression, can we use the knowledge that we derive from cell-free DNA to choose new treatments? And probably, yes, we can. We don't have the drugs necessary yet in all instances, but this will help us to guide us to develop the treatment that will be necessary to counter these resistance mechanisms. In the SIOPEN setting, we wanted to go beyond the single observations, and we want to really study in detail whether the fact that cell-free DNA actually sees early relapse is something that contributes for the overall treatment benefit, and whether this is something that will also lead to improved survival. To clearly document that, SIOPEN with the leads Lieve Tidgat and Sabina Taschner are proposing a randomized trial, Mona Lisa, for patients after relapse. And within this randomized trial, we will compare two patient arms. One arm where we will do standard monitoring as we do for any patient after relapse with imaging every 3 months. And then in the parallel arm, we will perform blood-based cell-free DNA analyses every month. And this will enable us to see if really, even after relapse, these cell-free DNA techniques are more sensitive, and whether this really contributes to improved survival overall for our patients. So this is the first trial that really prospectively evaluates liquid biopsies in neuroblastoma, and it will base clinical decision-making on the results of these analyses. We plan to randomize 150 patients. Dr. Titgat and Dr. Tashna Mandel reported that the first patients have now already been included, and we really think that in addition to the biological elements that we will learn, we will also importantly learn about quality of life and how these tools will be used within clinical decision-making based on the different tools that are being developed in this context. And finally, after having spoken for these uses for the diagnosis and follow-up, I also want to spend a few more minutes to think about how liquid biopsies can be used for research. In neuroblastoma, as in many other solid cancers, we know that not only genetic, but also epigenetic features are of huge importance. And we know that now that we can use cell-free DNA to study methylation, which is one epigenetic feature, in fact, methyl marks that are deposited on the DNA and linked to a suppression of expression. We can also study the fragment size of the cell-free DNA fragments and the nucleosome occupancy. First, with regards to methylation, in fact, the methylation patterns on DNA are closely linked to the cell of origin. And in other tumor types, it has been shown that methylation-based tissue of origin can be derived by studying cell-free DNA. And some teams have been using such approaches just to study methylation patterns to see the tumor type in a blood sample of patient with cancer. And this, of course, suggests hugely interesting possibilities in the future with regards to possible diagnosis. Dr. Burko and her team has gone beyond this and developed an approach based on what is called nanopore sequencing. So this is long-read sequencing that has been adapted for the cell-free DNA component. And in her recent work, she has shown that she can derive these copy number profiles and also correlate this with the disease status as indicated on the graph here, distinguishing patients at diagnosis, in remission, at relapse, and healthy individuals. And here again, the results as mapped on the bottom indicate a disease course that correlates with the clinical output and imaging. With regards to the research output, it is hugely intriguing to see that looking at these methylation patterns, differentially methylated regions can be observed that might be of importance for neuroblastoma, and I am sure her team will study these more in detail. And finally, another aspect that I think is of interest is that in neuroblastoma, we want to not just be interested in DNA patterns, but also gene expression patterns. But how can we infer gene expression from DNA? Here, we need to think about what I told you in the beginning, that in fact, the cell-free DNA that is released in the bloodstream is cut according to the position of nucleosomes. And the nucleosomes are not just positioned in an arbitrary manner across the DNA, especially across genomic regions called transcription start sites. Nucleosomes are either well-defined with really depleted regions or densely packed. In fact, across genes that are expressed, the transcription start sites have nucleosome depleted regions, whereas genes that are not expressed, the nucleosomes are densely packed. And this determines then the overall way that the DNA is cut when it is released from cells into the bloodstream. And several teams have made use of this to say that actually just the simple coverage across the transcription start site can help to infer expression of genes. In other words, a gene that is to be expressed, it will have a low coverage across its TSS, and a gene that is not expressed will have high coverage. And we've made use of that. So this is really experimental data from my laboratory, from my research team. And we've used an approach to capture the gene transcription start sites, and just built a model using machine learning to infer expression first with regards to genes that are known to be expressed or unexpressed in blood. And then we've used this to train our data set, and we get these graphs. The non-expressed ones are the blue on the top. The expressed ones have this dip. And then when we use this on neuroblastoma samples, it gets really interesting because we get these patterns. These are different samples, each one for a patient, and we can cluster the genes either with this high coverage across the TSS or the dip, and then this enables us to infer expression with either genes that are commonly expressed, that are listed here, or commonly silent. And so what we have done for now is compared the expression between MICAN amplified and non-amplified tumors in the plasma, and we really see differences in expression for some genes listed here. And so I think we will need to develop this further as we think more about how the DNA component is not the only component, but also expression is important in patients at relapse. An additional observation that is of importance when we study liquid biopsies, we can be interested not only in what happens in the cell-free DNA part itself, but also in the cellular component. And there, it is of importance to know that when we look at the blood cells, these are linked to hematopoiesis. And work published by the colleagues from St. Jude's, in fact, detected in cancer survivors that clonal hematopoiesis is present in a large proportion of these patient pediatric cancer survivors, and often also related to the treatment that the patient previously received. In this example, patient with Hodgkin lymphoma, where the signature related to the treatment with procarbazine could be picked up. So what we have seen in samples of neuroblastoma patients is intriguing, because starting at the bottom right, in the plasma sample of a patient at relapse, we picked up this mutation in the JAK3 gene with a low VAF. So this is a gene that can be altered in clonal hematopoiesis. When we looked at the blood cells in this same sample at relapse, we found this mutation at a VAF of 6.3%. However, this mutation was not present neither in the blood cells nor in the tumor at diagnosis. So this clearly highlights that there is evidence of clonal hematopoiesis. We can find this in up to 40% of high-risk neuroblastoma patients. We don't really know yet what this means. We don't know if this is linked in any way to disease response, etc. And it certainly needs to be studied further. So I hope I've been able to show you that indeed liquid biopsies are already in clinical use. And for neuroblastoma, we are using this for a molecular diagnosis. We're currently on the verge of developing this for disease monitoring, and we think that it is crucially important to implement these techniques in the context of precision medicine. And furthermore, there's a whole huge scope of further discovery for epigenetic analyses and proteomic analyses. So for clinical use, I think it's really important that cell-free DNA analysis can be performed at diagnosis or in the case of relapse. And this in particular, if no tumor biopsy can be obtained at the time of diagnosis, this might be small infants who are too unwell to undergo a tumor biopsy. Sometimes we can't find the primary tumor, and at the time of relapse, sometimes we just have MIBG spots and no tumor that can be sampled again. For disease evaluation, I think it's hugely promising. However, we don't have proof yet that detecting disease maybe 1 month earlier really will lead to improved survival for our patients. So we need to carefully advance step by step to think about how cell-free DNA analysis can be integrated into our disease monitoring and not overtreat patients who might otherwise not have disease evolution. And I think these are, again, hugely promising approaches for future research. I think in the overall research community, we will in the future think about integrating this in the initial staging for early response or clearance, probably also in the overall response criteria. And as I mentioned before, we will determine whether there is benefit in early detection of relapse. In this context, cross-border collaboration is hugely important, and the different collaborative groups, COB, SIOPEN, and others are already sharing data in this aspect, and we hope also to be able to integrate such data into databases such as INRG in the future. And of course, parents and patients support for all these approaches is hugely important. I want to thank for the work that referred to my own research, my research team and collaborators, and the fundings and the collaborators at the national and international level. And with that, thank you for your invitation to speak here and for your attendance. And I think with that, I am ready to take any questions. Thank you very much.
Okay, no questions. That means you're, okay, we do have one.
Would it be able to be used in ganglio? You mean ganglioneuroblastoma, probably. And ganglioneuroblastoma is a strange disease. As you know, it's a bit of borderline between aggressive tumors and non-aggressive tumors. So we would be able to see alterations in cell-free DNA, maybe, if indeed there are tumor-specific alterations. Some ganglioneuroblastoma don't have copy number changes, don't have mutations. In that instance, we wouldn't be able to see anything in cell-free DNA either. However, in other instances, ALK mutations might be detectable. So indeed, in that instance, we might be able to see something, yes. Looking to see if there are other questions. And a BRAF somatic mutation. And so if a BRAF mutation was known in the tumor, indeed, maybe it might also, again, it depends on what amount of cell-free DNA is circulating, maybe be found in the liquid biopsy. I see a question of whether we can use cell-free DNA to analyze the mesenchymal versus, I guess, neuroadrenergic or adrenergic neuroblastic cell states of tumors. I don't know if I can answer that question directly because the cell states are determined by core regulatory circuits, so it depends on expression. And so as I explained, we're currently trying to infer gene expression based on the DNA. So maybe that will be possible in the future, and that would be really interesting. However, we would have to be able to filter out one cell type from the other. So I think there's still a number of challenges here. I'm reading the questions for Mrs. Goldsmith. Shall I read the questions out loud, or can everybody see them?
No, please read them out loud.
Mrs. Goldsmith, who is asking about her son on his second relapse, with this MIBG spots, but liquid biopsy was negative. And at the third relapse, a lymph node, and there are results. So the question is, should he also do a liquid biopsy to see if there are different additional results? My personal opinion in this is that whenever a tumor is analyzed or whenever a relapse occurs, indeed, a liquid biopsy to study the genetic profile might be of importance. And it can be studied just with a small gene panel. A number of commercially available panels are available. I didn't go into depth with regards to this. And so I think it might be useful to guide treatment and to repeat this anytime. It might still be non-informative, but it might also be informative. So the idea would be to indeed repeat this upon each disease evolution. And I see another question from Mr. Lance, who is asking if I think that this testing will be standard protocol at diagnosis and throughout treatment in the near future. It depends on what near future is, but I think in the future, indeed, I think like in leukemia, where such analyses have now been standardized, it's very likely that this will be part of disease response and monitoring, but we're not quite there yet. Again, we need to be careful, and we need to be careful when we think about what we're looking for. There's a limit of detection, and we need to know also which tumor types release cell-free DNA and which tumor types don't. And then I see another question. If we have matched the liquid biopsies with biopsies of real metastases to know the cancer cell fractions, and indeed, that is really one of the interesting questions. What site of the tumor is releasing the cell-free DNA? We don't have the answer to that yet. And often we are limited because by definition, we can't take a biopsy of each metastatic site. But we're learning that what we see in liquid biopsies most certainly does not always correspond just to the tumor. And so we're learning to see also upon relapse, if the relapse comes more from a metastatic site or a primary tumor. I didn't include it in my presentation, but I can recount the example of a patient where the primary tumor did not have an ALK mutation. An ALK mutation at diagnosis was seen at a low mutated allele fraction. The patient unfortunately had intracerebral metastases and relapse. That doesn't happen often. And upon relapse, the relapse had an ALK mutation. So that enables us to infer that probably the relapse in the brain did not come from the primary, but from a metastases. So it's this kind of model that I think we will be able to develop more in the future. I see another question, with regards to tracking the metastases. Question with specifically to France, and I hope that indeed this will be in the near future in our European countries, etc. Again, I think we need to be careful with regards to interpretation of data and the limit of detection. And I see a question, is there a difference in cell-free DNA or CT biopsy? Not sure I fully understand. I think, the terms are used in the same way. Cell-free DNA means studying DNA in the blood, and ctDNA then means looking for the circulating tumor DNA part. Similar to the question previously from Mrs. Martinet from France, again, meaning once you want to look at one alteration, maybe make an amplification where we know for sure that that comes from the tumor itself. the question here is that some centers in the US are using this, others not. Overall, I think a number of commercial panels are available. We need to just be careful when a commercial panel is being used as opposed to the in-house ones, the one in shop or in other centers. The commercially available panels developed for adult oncology do not always target the genes of interest in pediatric oncology. For instance, I know of one or two that don't have the SMARCA4 gene on it. And SMARCA4 is important in neuroblastoma. So before going for a commercially available panel, it's certainly important to check that all the genes of interest that we think are of interest in neuroblastoma are indeed on the panel. I see more and more questions. I see a question, I hope I'm not forgetting any, from Mrs. Reynolds, who is asking, are there different types of liquid biopsies? Foundation one, that's one, indeed, that's one, it's one type of panel. And there are different types of analyses. It can be the Foundation one, the one from SHOP, other in-house panels. I cited the ones used in our institution. And indeed, using these for surveillance every 3 months might be of use. However, again, we do not know yet the sensitivity of this and if this correlates 100% with what might be seen on MIBG scans. I see a question which commercially available panels were inadequate. Now I'm incapable of remembering the name just now, but I will look that up and answer secondarily. But some adult panels do not contain the genes that are of importance for neuroblastoma, the SMARCA4 genes, etc. Another question, whether cell-free DNA, is it the most fragile or sensitive cells that undergo apoptosis and release DNA? I think it's actually the cells that go through the cell cycle quickly. So MYC and amplified cells, cells with a quick cell cycle, whereas cells which probably have a longer cell cycle. It doesn't always map out with the exact genetic alterations, such as ATRX or others, but there's probably not just the ones that are sensitive to chemo. It's probably also something to do with the cell cycle and other markers, and we don't have the answer to all of that yet. I think I've come to the end of the list of questions. I hope I didn't miss anybody.
Yes. Thank you so much for your time, your clarity, and for sharing your expertise with us today. This was fascinating. Your work continues to push this field forward and opens new opportunities, new possibilities for how we can understand and monitor neuroblastoma. To everyone join us, thank you. Your presence, your questions, and your commitment to learning are what makes this community strong. Together, we are building knowledge, connection, and hope. As we look ahead, our next session will take place on June 8th, where we hear from Dr. Giselle Scholler, who will share with us the BCC clinical trial update. We hope you all continue to join us in the coming sessions and as we bring more voices from across the global neuroblastoma community to share insights, updates, and progress. Until next time, take care, stay informed, and remember, together we can change the future of children with neuroblastoma. Thank you so much.